I think a couple of people have already joined. So I will just briefly introduce you and then you can start, right, sir?
Okay. Thank you very much.
So I welcome you all as a part of the Indian Society of Human Genetics Distinguished Lecture Series. This is for the first time Indian Society of Human Genetics has actually initiated a Distinguished Scientist Lecture Series. And this is basically to commemorate the 20th anniversary of first human genome draft and the 200th birth anniversary of Gregor John Mendel. For on this behalf, we have one of the most notable scientists in the field, Professor Aravinda Chakravarti, who is going to deliver his first inaugural talk of the Distinguished Scientist Lecture Series. And unfortunately, India could not participate in the human genome mapping program, but we had one of the most prolific Indian genomic scientists like Professor Aravinda Chakravarti, who had actually spearheaded program of human genome mapping program globally there. To the human geneticist, he doesn't need any introduction because he has already demonstrated his global dominance in the field of human genetics by playing a significant role in human genome projects. He had been one of the key architects of the human genome projects, HapMap projects, and the 1000 genome projects there. He had also been the former president of American Society of Human Genetics, which is considered to be one of the most powerful bodies of the human genetics in the world. And he had come several firsts in his career. So he had been the founding director and the Henry J. Professor of McKissick Methods Institute of Genetic Medicine at Johns Hopkins University from 2000 to 2007. Then he had also been the founding director of the Center for Complex Disease Genomics at Johns Hopkins again from 2007 to 2018. And he had been the founding editor-in-chief of the Genome Research and Annual Review of Genomics and Human Genetics. And he has also been one of the fellow of the most prestigious American Society of National Academy of Sciences, and also the US National Academy of Medicine. He is also the fellow of almost all the National Academies of Sciences of India. And I'm definitely privileged on behalf of ISHG to welcome you, sir, for this inaugural talk of ISHG's Distinguished Scientist Lecture Series. And we wish to look forward for your guidance and spearheading the activities of Indian Society of Human Genetics, as ISHG is planning to diverse its activities with respect to industry-academy interactions, initiate genome clubs in the schools and colleges, encourage charitable foundations and venture capitalists to invest in the genomics and genetic counselings, and also bring a white paper on the solutions and products or services that genomics can offer them. For all these activities, we really need your guidance, so therefore we will keep on bothering you from time to time. And one of the most unique part of Professor Aravinda Chakravarti is that besides his routine work on research, he has also published a book on cookery there, and the book is named as Not Everything We Eat is a Curry, a Bengali guide to Indian cuisine there. So it really opens up his versatility, not only with respect to genomics, but also with respect to the culinary art there. So welcome you, sir. And so now the floor is yours. So you can proceed with your presentation, sir.
Okay, thank you. Good afternoon to all of you. Onak, thank you very much for a very kind introduction. And thanks to all of you for spending part of the day with me and for inviting me to this inaugural event. I'm really very honored. I have unfortunately not really participated much in the activities of the Indian Society of Human Genetics, but it is true that I think this is the fourth event. I attended the first meeting of the Indian Society of Human Genetics in Mumbai that was chaired by Professor Lalit Sangvi. I attended then one in the late 70s in Chennai, I think one in the 90s in Delhi, And now you've invited me to this event. So again, thank you. Thank you very, very much. So I thought what I'm going to do today is try to convince you that regulatory variation as a cause of genetic defects and clearly of extreme human phenotypes are far more pervasive than what we focused on so far. I think often we use the word important. I'm not going to use the word important. I think in order for us to understand what the human genome contains, what the codes it contains, and how variation really leads to human phenotypes, their variants, I think we need a much broader perspective than we've had. And of course, there's no doubt that these perspectives wouldn't have dominated, at least my thinking, in the absence of the Human Genome Project and getting the first human genome sequence. So let's see. Why can't? Okay. So I'm going to speak just for a couple of minutes on our current very high level understanding on what's the nature of human disease today. It's really a sketch. Even though we've pinpointed genes for human disease, first using biochemical methods for a number of years, really the modern era started with position cloning. And I was involved in the identification of the gene for cystic fibrosis and identifying its major mutation, at least major mutation in European ancestry subjects called Delta 508. And this was important, at least to me, in beginning to think of not the old way in which we identified genes and therefore could study the phenotype, which is, somehow get to a biochemical defect or to a genetic lesion that would point directly to the mutant protein, but rather using mapping as a way to exclude many, many genes in a positional locus to try and find the allele. This is really the first time I presume anybody had, and we had used, to try and use association mapping to map a very common mutation, common at least in European subjects. Now, this is 1989. From then on, there have been numerous studies, and many of you have yourself participated in the identification of genes for Mendelian disorders. And this is sort of a synopsis that was published a few years ago. There are other such updates on the web that in Johns Hopkins and in Baylor College of Medicine, there's an entity called the Centers for Mendelian Genomics that was funded by the NIH to find almost every potential case of a genetic disorder that one could find, that one suspected to have a Mendelian origin, and to try and use sequencing to try and solve what the gene and its mutations could be. This is not the only contribution. There are many, many people, as you all very well know, including individual labs that have contributed to it. So here's a synopsis. So OMIM, that all of you know, which is the online version of the Mendelian inheritance in man, and really also celebrating its 50th anniversary, OMIM now has close to 8,000 phenotypes that are suspected to be monogenic. And of them, about 64% have a very credible gene that is associated with it. Sometimes this association, that is finding a gene with mutations and a disease, is very difficult because they're often very rare families. And there isn't the statistical evidence, but there may be additional functional evidence, other kinds of evidence that makes that link quite compelling. So here is what a typical experiment, for example, shown. This is using exome sequencing primarily, but sometimes genome sequencing. It's over 22,000 families, which were done as part of the Center for Medical Mendelian Genomics. And what this found, by the way, there's so many families, genes were not found in all of them, that about 1,430 of them were known genes. There were 470 phenotypic expansions, that is, phenotypes not suspected to be because of mutations in that gene, but so the gene is known, but the phenotype association was not. And there were about 1,800 completely new genes discovered. And as I said, this evidence is seldom statistical and is often based on only one family. And I think you cannot, it is good to be able to find these genes, but one has to be careful of how general one believes these statistics are, because I think these kinds of studies suffer from biased ascertainment. That is, we only pick genes that we believe, or rather I should say families that we believe are completely Mendelian, and these genes may have far broader effects. So this is what we have today at the bottom, that there are about a half genes for which we have no associated Mendelian disorder or a human disease associated. There are about 30% of genes that we presume, because of the distribution of genetic variants in large databases like NOMAD and others that you are aware of, for which there are no gene phenotype associations. We believe that they are embryonic lethal. And there's about 18% now. This is a collection of Mendelian phenotypes, remember, but if you look at the whole human genome, about 18% of genes, 20%, say 1 in 5 genes, have a clear Mendelian phenotype. So that just tells you that Mendelian phenotypes, which cause a high impact mutations or then arise from high impact mutations, are this 18%, and probably also these embryonic lethal genes. So that's only about half the genome, but they're half the genome. We have no idea what the phenotypes are, or whether the phenotypes are far more complicated, or whether they are due to far more, that is, they're far more complex reasons, and at least they don't have coding sequence mutations, as we know. Now, this is quite distinct to what we believe is going on in multifactorial inheritance. All of you are aware that multifactorial inheritance was a dominant theme in the early part of the 20th century because of work by Carl Pearson and others and his trainees. And even though the original hypothesis was opposed, that is, phenotypes due to the occurrence of many, many genes, it's quite clear that by 1919, Ronald Fisher, even though it was a theoretical paper, he proposed that these complex traits or multifactorial traits were nothing more than the additive effects of many, many genes. This, of course, was very, very difficult to prove at that point in time, even though there was evidence of multigenic inheritance in experimental studies. And this is Hermann Muller, really one of the leading lights of genetics, or has been. And Altenberg and Muller did a beautiful study. If you ever have a chance, you should read. It's on a phenotype called truncate wing that they called inconstant and modifiable. It clearly was not. It didn't Mendelize in the appropriate way in Drosophila. And they mapped it to 3 distinct genes. But today we know that Fisher was essentially correct. There is the Fisherian world, and there are many, many genes with additive effects. And I think his model is very important, for example, for predicting phenotype based on genome sequence and sequence variants, polygenic risk scores that many are currently investigating. But I wanted to delve a little deeper into what the molecular basis of such an observation might be. So again, I don't have to tell you, but I don't have to tell you the importance of the problem, but hopefully I will shed some light on what the molecular basis of these so-called multifactorial or non-Mendelian disorders may be. All of you know through genome-wide association studies, there are thousands of phenotypes with hundreds of thousands of associations that are known. These are replicated associations with a high degree of statistical certitude. And here we know that they are distributed all across the genome. It's not like there's half the genes that are involved. Almost every gene is suspected of being involved. And the common explanation has been regulatory variants. But what do we mean by regulatory variants? Well, the idea that regulation would be important actually goes back quite some time, but its importance in human phenotypes I think arose from some early observations and work that we had done in Herschbergs disease that I'm going to come to. And I'm going to point to this paper that was a collaborative paper that a number of groups, including my group, had published together with Eric Green. This is 2003, so this is just right after completion of the draft of the human genome sequence. The mouse genome had not still appeared. And even though this was painstaking work, the idea was that if you took a corresponding human locus, not only the coding sequence, a human locus, and you identified large insert clones and you sequenced them to great depth, and you compared them across many vertebrates. Here you see humans compared to chimp, baboon, all the way to zebrafish. What you would find, the details on the right-hand side, what you find is a high degree of conservation in certain segments outside coding regions. So by widespread here, I mean that this conservation is persistent. It's more than the coding sequence. By the way, coding sequence is only about 1.5% of the human genome. This is about twice that. And it is all over the genome. And it suggested that if evolution would have suggested, so this, by the way, the y-axis in each of these panels is the degree of sequence identity with human. Chimps are nearly identical here. This is a CFTR locus, by the way. So what you find is that here, for this gene, caveolin-1, that chimpanzees all, baboon shows a little bit less conservation, and by the time you get to zebrafish, you find very discrete elements that are conserved. And this conservation can involve both coding sequence, which is just a little bit, and a lot of non-repetitive, otherwise unique sequence. So we believe that these elements were involved, and this work came out because, or at least I was drawn to this work, and I participated in this work for specific loci that we were interested in the study of this really, for me, fascinating entity called Herschbergs disease. So Herschbergs disease, I think, has now turned out to be one of the models by which we can understand complex genetic inheritance. Very briefly, the genetic epidemiology of this disorder is that the disorder is associated with the absence of enteric ganglia. So there's a certain segment of the gut. This is a radiograph after barium meal in an infant that has Herschbergs disease. And you find a region of the colon that is very constricted, that in fact is a functional block, and dilation in its proximal regions. And this is because this is a transverse section of the gut in this cartoon, and here's the lumen, and what you find is there are these layers of muscle, and between these layers are these specific kinds of neurons, neuronal cells, ganglia, really organized as ganglia, and these large plexuses called myenteric and submucosal plexuses. And these are the ones that are missing in Herschbergs disease. There are many forms of the disease. There's a short, long, and a total chronic form. It has an incidence of about 1 in 5,000 in Europeans, about twice that in Asians, including in India. And originally we thought it was quite rare in Africans. Now it turns out with better surveys, that's not the case. It's about the frequency as in Asians. The disorder is not well recognized because these infants, if not recognized very early, a few days after birth, can clearly succumb to the disease. There's a lot of studies in the literature, including ours in the early 90s, that shows that this was a multifactorial trait. It has a 4% recurrence risk to siblings like other polygenic traits or multifactorial traits. It has a sex bias, but I was drawn to the fact that there are many syndromic associations that involve Herschbergs disease, telling me that there are many ways in which this disorder could arise, including from single genes. There's the Shaw-Wardenberg syndrome, which is one of deafness and pigmentary anomalies. That's a monogenic. It's a type 4 Wardenberg syndrome form. Down's infants, in fact, have 100-fold excess. So here it's chromosomal, and there are clearly other copy number variants, as well as more complex forms. And there clearly are animal models of the disease suggesting that discrete genes could, in fact, be the cause of Herschberg. So of all the genes, I think today I'm going to speak about only a few, of which the major one is the one with the largest number of mutations, coding mutations as originally discovered. That is, we had mapped, and the gene was identified first by Stan Lyonnais and And what we found instead is that there were many, many families that mapped to this region, families with multiple affected children, but in fact, there wasn't a coding mutation. And that was a reason for us doing the sequencing experiments with Eric Green that I told you about, searching for elements that were conserved outside coding sequence. And eventually from mapping, we found this element. This is a multiple conserves sequence. It's in the first intron. Here's exon 1 and exon 2. And it's about 150 nucleotides we'd first identified. And it clearly shows the maximum segregation in the families. We did in vitro work to suggest that this was likely a transcriptional enhancer. We knew that if we made transgenics with mice, that this piece of DNA, either a small one or a larger piece of DNA containing this element, that it was expressed in the right tissue in development. So we knew it could function as a transcriptional enhancer. And we later showed that this enhancer contained the sequence variant. So what this element had beyond being conserved is, it was a very common polymorphism. Its frequency is about 24% in European ancestry subjects. I'm going to call this red plus 3, and it was bound by the transcription factor SOX10, which is itself a Herschbergs disease gene. This polymorphism, by the way, again, is widely distributed in the world. These are control populations from the Human Genome Diversity Project. This particular polymorphism is rare in Africa, has a frequency of about 24% in Europe. And by the time you come to the northern parts of Asia, at least, what you see is a frequency of about 45%. It has an odds ratio of 4 for the disease, and it immediately told us that here was a necessary but not sufficient cause of Herschbergs disease that was non-coding, that clearly was a transcriptional enhancer, and that these enhancers could play a major role in disease. By this time, of course, many, many such examples are known. So what's the contribution of this enhancer that many people don't think is important, because they say, well, it has a small effect compared to coding mutations, and it by itself cannot lead to disease. But if we really quantify effect, here's a recent sequencing study, or recent 2 years ago, that we published. So we can find sequence variants that predispose to disease at a number of sites in genes. And they could be of various functional forms. They could be the transcriptional enhancers, like the one that I showed you, but the other ones that we've discovered. They could be the classical coding genes, as we talked about in the beginning. And they also could be large copy number variants, including trisomy 21 that I told you is involved in Herschbergs disease. So here's its frequency of cases and controls. It's not like controls don't have these so-called sequence variants. But here's the odds ratio. And what you find is the rarer the mutation, the greater the odds ratio. But enhancers have an odds ratio of about 4.5 together. But because they are so common in the general population, this is about 17% of controls, they explain a huge amount of risk. PAR is the population attributable risk. So here, enhancers contribute more to Herschbergs disease than do coding variants across all genes taken together than do copy number variants. Even the copy number variants have a much greater impact. So mutations with a greater impact are subject to selection. They are of lower frequency. But what we have to consider is how many patients arise from how many of these. So what immediately these enhancer discoveries told us is that if the enhancers have sequence variants that predispose to disease, and we already know of a number of genes that are involved in this disease, how do these genes relate to one another? So this sort of goes back to my previous slide, that right here, I've already listed more than 30 such elements. There were 34 elements, and the question is, at 30 elements at 34 distinct genes, and the question is, how do they relate to each other? Because we know individually many of them don't lead to disease. And what we found is that many, if not all of the genes belong to a gene regulatory network. This is the one that we figured out by doing work in my lab. This has mostly been led by Sumantra Chatterjee, and who's a senior research scientist here, and who's an assistant research scientist here. And this has been done both in cell lines, in mice, with mutant mice, and as well as with other experiments where you can knock down specific genes and look at their effects on other, for example, Herschbergs disease genes that we know, at least assess their effects by gene expression. So here's the gene that I told you about, RET. The red arrow here in the transcript says that they have coding mutations. We now know that there are at least 3 at this time of the publication, at least 3 enhancers. All 3 of them have sequence variants, common sequence variants that predispose to disease, but we believe that all 3, in fact, are synergistic, and the haplotype has a much larger sequence effect, for example, or deleterious effect. They have an odds ratio of 10. And these 3 enhancers, in fact, bind 3 distinct transcription factors. I spoke to you about SOX10 already, but the 2 others are retinoic acid receptor beta and GATA2. What's interesting is RET, in order for it to function in the early differentiation of enteric neural crest cell precursors into enteric neurons, requires another receptor. This is the endothelin type B receptor that we identified and positionally cloned many years ago, again, known to have coding mutations. It also has a number of transcriptional enhancers, but at least none that have common variants that are involved in Herschbergs disease. EDNRB and RET we have previously shown by work in both the Mennonite community that has a high frequency, high incidence of Herschbergs disease, as well as in mouse models, that these are very strongly epistatic. And now we understand the reason for the epistasis, and that's because EDNRB is controlled, again, by 2 of the same transcription factors, GATA2 and SOX10, not by RARB, but it requires a 3rd transcription factor called NKX2.5. So here are 2 genes that are very critical in the early development of the cells that will become enteric neurons that are controlled by at least 4 transcription factors, 2 of which jointly coordinate their action. And this in fact feature told us that the genes that are involved in such a multifactorial disease are not functionally disparate. They may have different functions, but the functions are coordinated inside the cell to produce a specific sort of biological task. In this case, to differentiate these cells into neurons. And if you fail in this task, then of course you don't have the neurons, you have aganglionosis and Herschbergs disease. There are many other genes that are involved. The ligands for the 2 receptors, GDNF and EDN3, a co-receptor called GFRA1, E3 ligase, but I'm just not going to speak about that. Hopefully what I've been able to tell you is that in this disease, there are many genes. The genes are not completely independent of one another. They're regulated in what's called a gene regulatory network, like the one I'm showing you. And this gene regulatory network connects the various components through these regulatory sequences called cis-regulatory elements or enhancers. Well, if you delve deep, you find that RET actually has many, many enhancers. I spoke to you about these 3 polymorphisms. The other red boxes represent other enhancers, also with sequence variants that are associated with Herschbergs disease. And of course, right now at this stage, when we've done a detailed study to identify additional enhancers in the RET locus. So here's the RET gene. Here's the original one that we've spoken about, the SOX10 binding enhancer and the variant. But the red here corresponds to many, many other enhancers that contain sequence variant, that are enhancers, sequence variants that are associated with Herschbergs disease, but they're related to each other by at least some domains of linkage disequilibrium. So they may or may not be new, may not be distinct. We believe they're distinct. Since then, there's been further work done by Sumanta Chatterjee and a prior master student called Kameko Karasaki, so here we've cloned out because of using a variety of criteria, something like, I think in this case, 27 different enhancers or sequences that act as enhancers in an in vitro assay in an appropriate cell line. But what I'm showing you is that if you now look at the difference in enhancer activity, right? If there's a sequence difference in an enhancer, then the two alleles must show some difference. What you find, some of them have marked differences, whereas many others have non-significant differences. So even if in this case, the blue is the non-risk and the yellow is the risk allele, the two alleles have a very small difference. So they could contribute to disease in a small way, but we cannot detect all of them. And what we find, if you look at all the red ones, I think there are 3, 4, 5, 6. Yeah, there are 10 distinct enhancers of red that contain sequence variants associated with the disease in which the two alleles have differential enhancer activity. This is just some further proof obtained by Sumanta to show that if in fact you delete these enhancers in a cell line model, you can show in almost every case you get. In one case with this E7, with this E4, you get an increase in RET activity, suggesting that the element might be a repressor, but all the others appear to be enhancers, because if you knock them out by CRISPR, what you find is decrease in the gene expression of RET. This points to an aspect of genetics that we need to understand, which is if a gene has coding mutations that lead to a disease, then specific kind of enhancer mutations that lead to the same gene expression changes are also associated with the disease, and transcription factors that bind that enhancer are also associated with the disease. This has led us to consider not a gene by itself, but at least what I call the transcriptional trio. And this trio says that expression of a target gene depends on this DNA looping. That's the current model, although other models are possible. And that these enhancers, like I showed you, they bind to specific transcription factors that then lead to controlled gene expression. And what you find if you have mutations in all of these factors, so here's the defect in a transcription factor enhancer and the target gene. The genetic effect in the first one is bound to be pleiotropic because a transcription factor is expressed in many, many tissues. The enhancer is generally tissue specific, and I'll come to this. And the target gene can be tissue specific or not. And this suggests that a transcription factor mutation is likely to be syndromic. An enhancer lead to an isolated case of the same phenotype, but the targeted gene could be either. As I briefly mentioned, if the enhancer leads to Herschbergs disease of an isolated form, SOX10, in fact, does lead to a syndromic form called Shaw-Wardenberg syndrome. And the target gene, of course, coding mutations can lead to, or in RET or EDNRB, can lead to both syndromic as well as non-syndromic forms. So if this is the case, and we have many studies in Herschbergs disease such as that going on, I think what I'm going to very quickly do is that if regulatory elements like this are really a contributor to disease, if not a major contributor to disease, as GWAS suggests, just doing mapping is insufficient. I think now what we have to do is do much more focused regulatory maps. And regulatory maps in this case, I really mean sequence-based in this genome world. That is, we have to know the precise sequences that are involved. We have to know the factors, that transcription factors that bind. We have to know what other factors that they associated with. We have to know the target genes, and we have to know what the target genes do. So I'm going to very, very quickly sort of go through just to save some time. Hello? Can you? Hello?
Yes, we can hear you, sir.
Yeah, but Your screen went.
The slides are gone. Yeah. But we're back.
Honestly, I don't know what's happening. So let me.
No problem.
Well, it is a problem.
No. It's okay.
Fine. Absolutely.
I just also wonder whether my system here just doesn't like GoToMeeting or something, because we don't use it. Okay, all right. So one of the ways that we've started this research program about 5 years ago is how we can very efficiently build sequence-based enhancer maps and all the details that I told you about in that transcriptional trio, and to try and build models that are predictive in the sense they can give us genetic predictions. So I'm going to use the term CRE for cis-regulatory element or enhancers individually. Again, I'm not going to go into the details because many of them, these have published data, and really this is really pioneering work by one of my postdocs who started this first in his PhD work with Mike Beer, and then continued with me, and now has an independent position at the Children's Hospital in Boston and Harvard, which is to use many kinds of epigenomic data that arise from transcriptional factor binding, say ChIP-seq, or much broader assays of open chromatin, like using DNA's sensitivity or ATAC-seq, and to find regions of the genome where, say, in any given experiment, you can identify these regions together with some negative regions and use machine learning that, by the way, has become very popular, but using machine learning to try and identify the specific sequences, the genome sequences that can act as these cis-regulatory elements. One of the advantages of such a genome sequence model is if you change the sequence context. For example, here is a C2A polymorphism. The wild type has C and the sequence variant is A, then the same machine learning model can predict its expression change. So remember, if in the multifactorial model, we need, what this means is if there are many variants that affect many enhancers of many genes, and each are contributing a small, then we have to identify all of those components. And here what this says is if there's a change in one of those components, like the sequence variants, then there is a method that Dongwon invented called delta SVM that gives us an idea, prediction, just like SIFT or Polyphen does. It gives a prediction as to what the impact on gene expression may be, and this has been validated in various experimental work, and the references are here. So I'm going to speak today just very quickly about the kinds of maps that we are building. We've already published the first major map of the human heart or cardiac enhancers, and this, of course, can use both data we generated on DNA's sensitivity with Greg Crawford at Duke University, together with publicly available maps, such as from the ENCODE project. The various reasons I could come to as to why we need to do this again, rather than simply download the elements from ENCODE. And that's because we can take various regions of the heart, like the right, or atria and ventricle and such, and we can have males and females and other kinds of biological variation that is important in genetics. And the reason for doing this is empirical maps are never comprehensive. It's just a snapshot of what we saw in any given cell or group of cells at any given point in time. The second is all enhancers don't have the same intensity. If you really look at this, this is a gene of great interest to us, the SCN5A, the most common sodium channel in the heart. You'll find enhancers that have very low activity as well as enhancers that have very high activity. And so, as you can imagine, the larger peaks are easier to capture in an experiment than the smaller peaks that vary. So we need multiple data sets to be able to look at the signal intensity of enhancers. Sequencing depth also randomly varies across the genome, the protocol differences. And also chromatin state is not a fixed quantity, it is dynamic. And so we need many observations. It's the difference between a still photograph and a movie to understand open chromatin. So in this first iteration, we did 5 samples.
We defined cis elements by 600 nucleotide elements, and we classified them, or we described them by sequences of 11 MERS. That is, it's this population of 11 MERS within the cis regulatory element that defines it rather than a simple linear sequence. And that's because transcription factors bind the DNA by various kinds of associations. Because we have a model, we not only found 160,000 such enhancers for the heart, but we also could predict many more. The predicted CREs, in fact, have weaker signals, but they have the same sequence motifs. They're much more cell type specific, so they're far more difficult to identify. And they also map to repeated sequence DNA, which is why, depending on how you do the analysis, you may or may not find them. We know that these CREs overlap conserved elements. The observed ones are highly conserved, about 97%. Even the predicted ones are much more than randomly identical, meaning randomly conserved or identical, and they also overlap cardiac relevant open chromatin. And we've identified by the whole set in the heart many cardiac transcription factors or transcription factors expressed in the heart belong to many families that we already know are involved. We've now done this for all of the 5 regions in multiple males and females. And I don't want to describe that work, except just to tell you that how they can be used. So here's a specific gene. This is SCN5A, the major sodium channel of the heart. In fact, it's an ion channel that is extremely important for the maintenance of cardiac rhythm. So these are similar kind of assays. We identified many regions, in this case, by this open chromatin analysis. These are potential enhancers. It just tells you the relative intensity of the enhancers. We found sequence variants in many of them. We've used other data, for example, from the GTEx project to try and predict what the expression will be based on the models that we have versus what we observed in heart tissue from GTEx. And here what you find is they in fact have very good agreement. You can look at this paper we published a couple of years ago. And what we find is from the whole collection, from these, we can identify here, highlighted in yellow, specific variants in specific elements that have many other properties that we expect for a cardiac SCN5A enhancers. Either they bind some cardiac protein, although they have high delta SVM scores, or that they overlap a known cardiac open chromatin segment. And so we think that in this way, we can do this on a gene by gene basis. But if we do so on a gene by gene basis, what will it overall tell us? So I think where we are going towards, or at least my laboratory is going towards, is we are going to use the genome and its sequence and its sequences across multiple individuals, and these kinds of epigenetic maps using a TAC-seq or DNA-seq from multiple tissues to try and make these detailed maps. We're going to use these maps to predict gene expression in individuals, not overall, but in individuals. And from this, we're going to try and predict their effect on an overall phenotype, and thereby understanding all the components that are involved. So here this suggests that the kind of work and what we've generated, some of which are generating in the lab when they're absent, others can be found from ENCODE. We do very stringent, this is the second reason why we are generating these kinds of data, because we find some of the published data are not of the greatest quality to do the kinds of work that we are doing. But once you have the map, which is after quality control, after QC, which is the kind of maps for the adrenal gland, the left ventricle of the heart, for arterial samples and kidney samples, all of which are important for our understanding of blood pressure, for example, or other disorders, that we can use it to understand the full repertoire of genes, their enhancers, and their transcription factors. Again, today, for lack of time, I'm not going to tell you that this peak quality analysis, again, based on some new methods by Dongwon Lee, is very, very important, because it tells us or gives us ways of trying to identify the subset of the data that has the greatest reliability. And this, of course, has turned out to be very important because it allows us to make very, very specific replicable predictions. So here's at the end, I'm going meaning this of this study, this is what we have. These are the enhancers or cis elements that we've identified. In this case, we defined a somewhat longer window. We found that these 2 kb windows give us far greater reliability in looking at what we consider is the enhancer. And here are the numbers that we found from, in this case, 4 tissues. They all have somewhere like 110, 130,000, 140,000 enhancers. Here's the average length, and here's the genome coverage. But if you take all 4, for example, in trying to understand blood pressure, obviously there's redundancies. That is, some of these enhancers are present in open in multiple regions. They still comprise about 20% of the genome. So as opposed to many Mendelian disorders, here we find that much of the genome will have elements that control its transcription. And I'm not saying all of the 20% is important, but the 20% is highly enriched for elements that do control expression. So here is one of the interesting features. If we look at these elements and their predictive power of, say, blood pressure or the heritability of blood pressure, we find a very sort of unique situation. So here are the 4 tissues, and this is the kind of complicated Venn diagram we can build now. And what you find is much of the variation that enhancer variation that contributes to blood pressure, much of this variation occurs either from individual tissue, adrenal only, artery only, kidney only, or the heart only, or it arises from the 16.5% that is common to all 4. So there appears to be 2 kinds of enhancers, those that are tissue specific and those that are ubiquitous, at least with respect to the ones that I've shown you. So if we take all of these CREs, how much of blood pressure does it explain? So for this, we've taken all of the variants that are polymorphic from the 1,000 genomes. We've taken, in this case, we've analyzed the UK Biobank summary statistics for systolic and diastolic blood pressure. And we've tried to do this partition heritability analysis by looking at only the functional regions that we have defined. And what we find here is that in this data set, the SNP heritability, that is really the additive portion of the heritability that sequence variants across the genome can explain, is about 14%. This is about half the total heritability of blood pressure. And what I'm showing you at the bottom is the results for systolic blood pressure. So if you look at adrenal, heart, artery, and kidney, here is the proportion of this heritability that they explain, which is substantial. And if you look at these 4 tissues taken together, we can now explain about 65% of the blood pressure heritability just from these 4 tissues. Of course, these tissues were chosen because they are thought to have a very high involvement because of physiological reasons. This is significant enrichment, and these are all highly significant so far as p-values are concerned. And we are now trying to do this with many other data sets to show that these tissues and their specific enhancers are very specific explanations of blood pressure variation. So I'm going to end just with a couple of comments on at least to tell you of my thinking of what is the scope of regulatory defects in human disease. First, I think making these maps, making better maps, just as we've seen with the sequence and sequence variation and others, I think better maps is important. The maps need to be quantitative. The map need to have very high quality. And even though there are many large projects such as ENCODE out there, and their results are very valuable, I think the analysis together with urine analysis of features such as sex difference and such is very, very important. So One of my thinking and the thinking of my laboratory is, even though we rely so much on data, we cannot rely on analysis alone. We have to generate some of them just to get far greater specificity of the results. Just like the human genome sequence is great, but of course is not useful to your particular case, particular patients, where you've got to sequence specific genes of interest. So one thing to point out is we can, that is DNA sequence, DNA's hypersensitivity and ATAC-seq are not the only ways of doing it. We can create maps of other kinds of epigenetic marks, histones, DNA methylation, and others, and analyze them in exactly in the same way, eventually concatenating these maps and finding out where they have commonalities and what the commonalities mean to try and develop a quantitative epigenetic regulatory code for every phenotype of interest to us. The second is, I think some talk about enhancers only in the context of multifactorial or quantitative traits and not for Mendelian disease. We've shown this before. This is again a case of Herschbergs disease that sort of changed our thinking. Here's a family that we know of that have 2 kids with severe disease. And they arose from 2 parents. These are 2 siblings, one of whom had very mild disease, a very ultra short segment, and the other one was clinically unaffected. In this family is segregating in nonsense mutation in RET, and it arose in the grandfather, was inherited by both daughters, and also by the 2 severely affected children. What's happened in this case is that both the fathers of these children, grandchildren, have contributed a haplotype that contains that enhancer mutation that I told you about, that RET+3 SOX10 binding site. So the difference between these 2 parents, by the way, nothing in genetic is of, I don't think that, or I should say, that not every trait, even Mendelian trait, is fully penetrant. So there is stochastic change, some variation, that these individuals, in fact, have only half, 50% RET. And that leads to mild disease in one and no disease in the other. But these individuals produce even less RET because the enhancer mutation on the other side decreases wild-type activity even further, so that these individuals have less than 50% RET, which is, I believe, what the cause of Herschbergs disease is. We've written in this paper and studies by Ashish Kapoor, whose name you also saw in the studies of SCN5A, that enhancers next to important developmental genes are very important modifiers of the expression of Mendelian changes or Mendelian mutations. So for example, a regulatory element that is next to, let's suppose, a mutant allele, if the regulatory element can decrease the activity of the mutant allele, you're going to have a far milder phenotype than if you have a regulatory element that decreases the effect of the wild-type allele. So there are many possibilities of enhancers that are linked to a mutant copy that can increase or decrease its expression, changing the gene expression of the target gene and therefore the expression of the phenotype. But overall, except for Mendelian traits, when we look at multigenic traits, we have to look at the problem in a much broader way. Again, Herschbergs disease has been a useful model in this. So here I'm showing you a multifactorial trait with many, many genes, each of which have different enhancers that have sequence variants that change the gene expression. And in an individual, that gene expression could be above the mean in this case, but in another individual could have a different value. These gene expression changes independently don't cause disease. They may in some circumstances, but usually don't. Their effects get united through a gene regulatory network, such as the one for RET and EDNRB that I showed you. So clearly cells have ways in which they monitor gene expression of individual genes. They assemble these in machines that may be called gene regulatory networks that ultimately produces an output that in fact changes cell behavior and function. For RET and EDNRB, the altered cell behavior and function is cell loss or the loss of cell proliferation. And it is this cellular event or the cellular phenotype that leads to disease. If this altered cell behavior, for example, is mild, you get mild disease. Otherwise, you get probably severe disease. So in genetics and in genomics, we are on the left. We are trying to quantify not only this genomic component and variation component on the left, but also this gene expression component. And if we can do that, as I showed you in Herschbergs disease, we can build gene regulatory networks through now very powerful single cell and CRISPR-based mutagenesis. And I think one of the lessons we geneticists should learn, as we have learned by studying Herschbergs disease, is we ultimately, to understand disease, have to understand the cell phenotype and really understand the underlying cell biology of the disease as well. So one should look at multifactorial traits in a some bit more graded way. And now we think that disease, extreme phenotypes in these, blood pressure is not a disease, by the way, it's a quantitative phenotype, but clearly extreme untreated blood pressure leads to clinical hypertension that is can be very serious. And we think that what happens in any given trait is that the disease occurs through a failure of many, many steps. But the many steps that it fails in lie in a cascade. For example, compromised transcription factor and enhancer function has to be right. If it's not, it may or may not change local chromatin structure, which can change, but that does not have to change gene expression. That does not have to change protein expression and down the line. What I mean is one has to have proper wild-type function for all of these factors before one gets a normal homeostatic wild-type phenotype. But genetic defects in any one of them is insufficient. We get a phenotypic response when all of these factors fail. And one of the things we're now doing with my collaborator and colleague here, Jeff Buka, at NYU, is now we're using big DNA technology, that is synthetic biology, where we can construct these very large loci, such as for Rett in mice, that contain the human locus, are deleted from the mouse locus, can contain all this regulatory variation so that we can study all of these changes to understand how multifactorial diseases come about. I'm going to end with these acknowledgements. I pointed out work of Dongwon Lee and Sumantra Chatterjee. Prabhu Mathialagan did much of the epigenomics, the data collection and the analysis. Ryan Fine is working on this big DNA of Rett and in mouse models. I mentioned a lot of work of Dongwon Lee and recently from his student Shong Han. And these are previous collaborators, many collaborators that I mentioned. I mentioned Greg and Ashish, but Santi Ganesh in Michigan on a blood pressure studies and prior studies with Mark Halushka and Dan Arking at Johns Hopkins. Of course, I am indebted to the many, many community genomic resources that have changed our work and for the grant support. So I'm going to keep this here, and I'm glad to take questions if, in fact, you still have the patience to do so. But I apologize for this delay.
Bonek, can we ask?
Yeah, so now it's open for discussion then. Yeah. Is it Rachel?
Yes, it's me. It's me.
Yeah,
Rachel.
yeah, you can.
Yeah, that is a really nice talk. Just that single gene disorders to those multifactorial. In fact, most of them could be multifactorial if we included the regulatory elements. Dr. Chakravarti, you there?
Yes, I'm here. I think there clearly are phenotypes that are Mendelian, but I think they are more the exception than the rule. And as you well know, we have struggled with trying to explain variation in patterns and variation in expression of the phenotype, what do we call expressivity, even for Mendelian traits for a long time. And I think this is one way to explain that, but it clearly is the major way to explain what happens for multifactorial traits.
Yeah, I think this could rather be the rule than the exception, is what I honestly believe.
Yeah, I believe it's the rule rather than the exception, simply because there's more regulatory, there's more of the genome reserved for regulation than it is for the coding sequence. And as well as most phenotypes that anybody has measured, at least if you measure it in a quantitative way, does show variation.
Yes. And now these, what you talked of, are the cis regulatory control or elements. But, I'm sure there will be an equal contribution or a greater contribution from trans regulatory elements as well.
Yeah, so I believe I mentioned that we obviously, not obviously, we are also studying all the trans factors or the transcription factors and other chromatin regulators and other kind of trans factors that are responsible. It generally is the case that the trans factors are highly conserved. So coding sequence changes in them is limited. Also, transcription factors generally are not tissue specific. They are actually broadly expressed. And it's really the combination of transcription factors that brings uniqueness to a tissue. So I'm not saying that transcription factor variation is not important to disease, and it is an important thing to study, and we are studying. I just think that cis variation is going to dominate. And it has nothing to do with cis versus trans. It has to do with the fact cis elements can suffer a lot of variation, much of which doesn't do anything, specific amounts of which change gene expression, but that perturbing transcription factors is more difficult because they are much more strongly regulated.
Yeah, now with all the small RNA regulations.
I'm sorry?
Small RNA regulations like the micro.
Clear. I mean, exactly. I think there are many other components to regulation. I think what we have to, however, remember is like coding sequence is about 1.5% of the genome and a very large mutational target so far as its effect is concerned. Cis elements are about 4% of the genome and also a very big target so far as variation in phenotypes are concerned. Small RNAs and others are a far smaller component. I'm not saying they have very important physiological roles, but as a mutational component, current evidence is not great. That doesn't mean that it will not change.
Yeah, as a mutational component, we don't know much. Yes, please go ahead, someone else.
Are there any further questions?
Yeah, Moina, can I?
Yes, stand up, yeah.
Okay. Hi, Professor Chakravarti. Really brilliant talk. I think, very, very contemporary and really full of information. So I had some difficulty in between, so I'm not sure whether you had covered this or not. But many of these diseases, whenever we talk about these epigenetic modifications, we have an issue whether this is an outcome or really a cause. So two questions related to that. One is that what do you think is the trigger for these modifications, number one? And second is that how you are so sure that this is actually, the one which is causing, or rather than it's being the outcome?
I think the reason why we know, let me answer the second part first. I think the reason why we know that they are causes rather than effects is that we are looking for sequence variation that is in the germline. And we are comparing individuals who have one kind of variant versus individuals who have another, the alternative variant. And so that itself tells us that there are host differences that lead to all the succeeding differences that we see. So I think we have to distinguish two kind of epigenetics here. They're clearly epigenetic changes that are outside the DNA sequence. But I'm speaking about epigenetic changes that are due to genetic changes in the epigenetic machinery. And so I think by the usual criteria of comparing individuals who are of one kind or another, we know that that's the cause. The second is, we have and others we've created, mouse mutations with no other change that we know of, but clearly in isogenic lines where you can create such effects. So we know that by that same criteria that these are effects. I think ultimately you may say that, well, if I think for Rett, for example, a Herschbergs disease, we expect that that's what we're going to see. We see it in cell lines, for example. If we can block the enhancer, then you can block the gene expression, and you at least find gene expression differences. Cell lines don't have phenotypes, but they do have, for example, this proliferation phenotype. So there's a analogous cell phenotype that is consistent with everything else. But ultimately, we have to build these complex models, which is why, these large models where, as I told you at the end, we will have no mouse endogenous gene. We will have only the human endogenous gene. That is, at least for Rett, we know the mice do fine. And then we will create nothing else but these enhancer mutations as we have. And we will see what the phenotype is. And we will see where or how much of the phenotype we get. That is, it is possible we will not get Herschbergs disease, but we will still get altered cell behavior. But the cell behavior may not be sufficient by itself to lead to Herschbergs disease. I'm not saying Rett is the only cause of Herschbergs disease. There are other genes. But I think those are other kinds of mutations we will have to bring in. So,
right, yeah. Can I ask one more question, Moinak?
Yes, please go ahead.
Okay. So, I mean, just to continue on this, while we, thanks for clarifying that. In many of the complex diseases, we talk about these, environmental changes which actually influence DNA methylation. And then we also know that they are likely to influence the transcription. Sometimes we know that there are MQTLs, which actually are genetic changes, which can influence these, methylation changes. So what would you think about their role? I mean, I understand Herschbergs is a very different category, but if we talk about true complex, like real complex diseases, like say diabetes or cardiovascular disorders, or say blood pressure for that matter, what will be your take on that?
I think the increasing evidence, it's not known for blood pressure, but the increasing evidence for a certain number of phenotypes is that the environmental effects on a phenotype actually occurs through these enhancers. These enhancers are, in fact, sensors. I think I pointed out this transcriptional trio of the transcription factor, the enhancer, and the target gene as a trio. I think this is sort of an important concept, that meaning we've known of these components for a while, but I think they act together as a module. We know, for example, that environmental factors, and it depends on the environmental factor, but there are two extreme cases, or two case examples. One is, all of you know of the case for thalidomide embryopathy. So thalidomide, once it diffuses into cells, in this case, specific cells during early development, what it does is it inactivates a transcription factor called SAL4. And if you inactivate SAL4, it fails to turn on certain genes that are important for limb development. That's why you get this. The reason why this is instructive is if you have mutations in SAL4, which in fact do occur in some rare families, you get the same embryopathy. So I think this is a clear case where an environmental agent directly affects a transcription factor, which then can change development or a phenotype drastically. The other now is very well known in the case of, say, oxygen. Oxygen is sensed by cells, as you know, through the IF1 and other pathway. And oxygen sensing occurs through early chromatin regulators, like some of the KDMs. And so oxygen, again, diffuses into cells. These transcription or chromatin regulators, in fact, sense them. It changes the activity of those regulators that then can change the physiology of that cell. So I fully expect that, I don't know about all environmental factors, that many environmental factors will have their effects either through the transcription factor or through other such factors that are going to affect gene expression through the enhancers. So I don't doubt for a second. I think the idea of doing genetics in this way is to find the components. I'm perfectly capable of accepting that in many individuals, the same components will be perturbed with other factors. Right. It doesn't have to be a mutation. It could be other environmental factors.
Right. Thank you very much, Prof. Chakravarti. I'm done, Moina. Yeah.
There are plenty of questions that are coming up. So, in fact, we will try to conclude by one or two questions itself. I have a quick question. It has been a phenomenal talk because none of the geneticists actually try to focus on the regulatory elements or the enhancer elements there, because that's like a risky domain of activities unless you have enough money to work with. So I just want to have a query that, do you find any relationship with respect to enhancer elements upstream to the start site with respect to phenotypic variations or the age of onset?
Yeah, so I don't know about age of onset. I mean, we haven't studied age of onset, but almost all of the enhancers we are looking at are within 25 to 50 kb upstream. They, of course, can be within introns, particularly the first introns. I suspect this is particularly the case with many neural genes. Usually genes expressed in the brain, they have an excess of genes with very large first introns. And I suspect that many of them have these kinds of regions of open chromatin and enhancers. But most of them are 25 to 50 kb away. There are some classical cases, as you know, involved in Pierre-Robin sequence or in postaxial polydactyly that are even a megabase away, sitting in an intron of another gene, completely unrelated gene. And they will in fact, they will occur. But I do believe that if one considers, say, phenotypic variation of Mendelian diseases, then I think searching upstream of that gene for specific enhancers that can modulate the expression and therefore at least those with variable expressivity, I think will be very fruitful. This has been shown classically in the last 20 years, mostly by Veronica van Heiningen and others, that there are enhancers of BAC6 that in fact have mutations that change BAC6 expression in development and lead to variable expressivity of aniridia. So I don't think that everything has to, that is, our focus is on both, as you saw, a very rare disease and as well as common disease to understand the genetics. But any of you who are looking at any other, say, Mendelian disease that has expressivity changes or age of onset, as you mentioned, then yes, I suspect that it will be a that you can do.
Yeah, so I'll pick up another question from Sudhanand. Some studies correlate enhancer activation to transcriptions of enhancer RNAs from these CREs. How do these enhancer-derived RNA regulate the expression of genes involved in the multifactorial disease?
Yeah, so we don't know. That's a very good question. We don't know. Enhancer RNAs, are obtained from some enhancers and not others. By the way, enhancers are just not one thing. I described them in a very simplistic way as binding factors, but we now know many kinds of enhancers, and even the same enhancers has different roles. So the reason why I think the study of enhancer RNAs in human disease, except in cancer, hasn't proceeded very much is you have to, if you concede the observation that many of these effects are tissue specific, you have to look at this in the tissue of interest. And that, of course, is a challenge and often impossibility. So the advantage of studying sequence variation, germline sequence variation, is you can study it in almost any given tissue, right? I mean, this is just DNA obtained from blood. So there's no doubt that those kinds of things are happening, but I would separate the mapping of elements, transcription factors, genes that are responsible for a phenotype from its functional dissection. That will no doubt require animal models, cell models, and even engineered models.
Thank you, sir, very much. And will you be interested to take one more question? Last question?
Yeah.
Yeah, so this is the last question by Ajay Deepak. He asked, considering the fact that expression of gene can be influenced by multiple enhancers interacting with the gene promoter, how much variability in gene expression genetic variant in one of the enhancer can cause?
Yeah, so I think we also have to distinguish between what these enhancers do functionally from how we study them operationally. Okay. And what I mean is, I know that there's all this debate about each enhancer effect being small. Yeah, a single enhancer probably makes a small contribution if measured in a marginal way. But to me, that's not how the enhancer acts in the cell. In the cell, for any given gene, there are two copies. It has a series of enhancers. The enhancers has whatever sequence variation. They act together, like I showed you in the transcription trio model, and it is a combined effect on gene expression that is important. When we do an experiment, we can measure it piece by piece by piece, but the biology doesn't happen piece by piece by piece. So I think what we have to think about, irrespective of how we detect, which could be individually, that what is important is the total effect of the haplotype that contain all of these in the individual. And I think that's what our approach has been. And those effects can be substantial.
Thank you very much, sir. In fact, it had been a very exciting talk, and there are plenty of questions there from the various attendees. I thank you to take time off from your activities, from your work, and that to come on online, that in the early morning. Thank you very much for giving an exciting talk, sir. Thank you very much.
Well,
And I
thank you
thank-
very much for inviting me. Wish you all very well. And if you need answers to specific questions, I'm glad.
Sure. So I'll ask all the persons who attended the talk to kindly get in touch with Professor Aravinda Chakravarti to raise the queries in case if you want to resolve it further. Because we can keep on going on for almost an hour. I think that the queries are too many. So I think it's already, we are 6:30 here. So thank you very much, and thank all the attendees who have attended through the various channels, whether through GoToMeeting or through YouTube. So we are extremely thankful, sir. With these words, I possibly call a day today. And once again, thank you all for attending the online talk.